I'm Dr. Monty Powell from City of Hope, and this is On the Edge of Breakthrough, Voices of Cancer Research. Each episode, we bring you the minds behind the science, the stories behind the data, and the breakthroughs that could change everything. Let's dive in. Welcome back, everyone. I couldn't be more excited to have our guest today, Dr. Leo Wang, who's actually the head of our Alpha Clinic here at City of Hope. He's the head of our gene therapy program here at City of Hope. And on top of that, just a world-class, world-renowned pediatric oncologist. Leo, welcome to the program.
Thanks so much for having me. It's a real pleasure to be here, Monty.
You got it. So, I'll tell you, we usually kick off things by really talking about everyone's origin story. What sort of got you into the field of oncology on the whole? Can you give us a little primer on your background?
So I grew up in upstate New York and went to public school, and then very early on discovered that I had a huge love of science and started doing research in a research lab at the University of Rochester when I was in middle school.
In middle school? Wow, that's early.
It might not have been legal at the time, but it was a lot of fun, and I realized I was both really good at it and really loved it, and so decided that I was going to devote my life to that. Ended up going to MD Ph.D. school after undergraduate and got my MD and Ph.D. at that time in developmental immunology. So I was studying B cell development. Those are the cells that make antibodies. Those are a particular type of immune cell. And then from there, when I was finishing up medical school, decided I really wanted to do pediatrics. And that really was just born of a love for kids and coincidentally had my first kid at the same time. And so it was sort of paralleling what was going on at home with what I was seeing on the wards, and really fell in love with that patient population.
Interesting. We're going to get back to pediatric oncology specifically, because I think it takes a certain kind of person to be able to see, children affected by cancer, right? But before we go there, right, you have this really wild story to tell us regarding your siblings, right? All three of you are in different fields. Right. Can you give us a little preview or primer on what they're doing these days?
Yeah. So, I don't know if that's that wild. It's it's
pretty wild. It's pretty wild.
So I'm the oldest of three. And we all grew up in upstate New York. My middle brother is 2.5 years younger than I am. And my youngest brother is 12 years younger than I am. And so, in Chinese culture, the family names are often, you start with your last name, right? Because that's the most important thing is the family that you're from. And then it's a 3-character name. And the first character of your first name is actually shared generationally.
Okay.
So I'm Wang Li De, okay? And my brothers are Wang Li Hong and Wang Li Kai. So we all have the Wang, which is our family, and then the Li, which is sort of our stratum within that larger Wang family. And then the last part, that's where you get to be individual. So my middle brother is Wang Li Hong, and he is probably the biggest Chinese pop star ever.
So this is kind of a big deal, right? Yeah. This is kind of a big deal.
Yeah.
You got to tell me how that happened. I know I digress a little bit.
No, yeah. He did not work in the lab when he was in middle school. Let's put it that way. But yeah, he's been very talented musically. We all grew up playing instruments, and music was a huge part of our lives. But he had a particular talent for it that was apparent very early on. But I think that, he wouldn't have gone on to do what he did, except serendipitously in between his high school and college years. That summer, my maternal grandfather got sick, and he lived in Taiwan at the time. So my mother went back to Taiwan to care for him. Because my brother had graduated from high school and didn't have anything to do for the summer, he went with them. And every day they would go between where the apartment was that they were living and the hospital was where my grandfather was staying. And they'd walk by this, strip of basically shops. And there was this one particular building that had this very interesting facade, and they didn't really know what it was. So one day they went inside. It turned out it was a nightclub. And Li Hong was like, well, I'm here all summer. I had nothing else to do. And, maybe I can audition for the house band or something like that. And he didn't speak any Chinese at the time. So my mom went to the manager of the club and said, hey, my son is a musician. You mind if he auditions for your house band? And they're like, well, normally we'd have auditions for the house band on Thursdays or whatever. But this Thursday, we're not doing that because there's this national talent competition. It's Star Search back in the day. It's the winner gets a record contract. So whatever. And Liam was like, oh, I'll do that then. And so he entered this competition And after his first round, people started calling him. They're like, hey, we saw you at this thing. We'd like to give you a record contract.
So he was in this American Idol type of deal.
Yeah, exactly.
More or less, right? The
Taiwanese Idol, I guess.
Wow. And your younger brother, tell us about his story. Is he also in medicine or?
So Li Kai is the smartest of the three of us by far.
Okay. Okay.
And he was this kid who, when he was like 7, was just like making these observations that just blew us all away. And he's always sort of, because of that, been able to see the world in ways that I think that are not open to the rest of us, and very quickly figured out how to sort of, create his own path. So he actually stopped going to high school in the middle of high school. My dad happened to be home, which was rare, but he came down and he's like, aren't you supposed to be going to school? And he's like, I don't really think there's any reason for me to go to school. Like, I already know everything they're going to teach me. And my dad's like, okay, that's fair. Yeah, I agree. You don't have to go to school anymore.
Wow, what an open mind. Okay.
Yeah. Anyway, he super smart, got a perfect score on his SATs, and ended up going to MIT. Thereon, he went to work on Wall Street and was a very early quant trader. You know what that is? A quantitative trader. That's they wrote computer algorithms that would take advantage of very slight mismatches in markets.
Okay.
So in the time that it takes to register a trade between two, whatever it is, commodities or stocks, there's a very small window where there's a mismatch in the price between the seller and the buyer.
Okay.
And so if you can take advantage of that, even though there's maybe a couple of cents mismatch in the price, you can make a ton of money if you slip in and intermediate between that discrepancy. But it requires you to be very, very fast, right? Okay. Human beings don't have the speed to do that. So
and he's like writing the software.
Yeah, it got to the point where what actually was really important was not just the speed of the algorithm, but how close the computer was to the exchange.
Okay.
So if you put a mainframe like right on Wall Street, then your lag was like much shorter than everybody else, and you could make millions and millions of dollars.
I feel like we've got to have a podcast. I'm looking at my producers right now on the Wang family. I mean, I think this is really, we're getting a little bit beyond scope here, but I'm really enjoying this. That's wild. Wow.
Yeah. So he did that for a while, made a bunch of money, realized that was not making the world a better place, and so quit. And then actually worked in the entertainment industry for a while, then realized that was not really making the world a better place in the way he wanted to. And so quit that and actually went to go work at UCLA in a genomics lab, and doing, genomic sequencing to try to help people identify patients who have, undiagnosed disorders, like what the genetic mutations are that underlie that.
Just amazing.
Yeah. So that's my family.
Incredible.
Yeah. Are we out of time? Thank you for tuning in.
There you go. No, it is just wild to see 3 siblings with such divergent interests, but also incredibly successful in their own right. But we're going to shift focus back to you for a second, Leo, if that's okay. And one of our first podcast guests was Saro Armenian, who is the chair of pediatric oncology here. One of my best friends here at City of Hope. We actually started the same year here about 20 years ago now. Bring up Saro because he had this just amazing, touching story of, what got him into pediatric oncology. And, we didn't get enough time on that podcast to devote to how you sort of make it work, right? I mean, seeing kids with cancer, especially having your own children, it's got to be, on some level, so, gut-wrenching and emotional. How do you sort of deal with that element of the job?
The reality, I think you're right, that pediatric oncology has to be a calling. It is without a doubt the greatest honor and the most humbling honor of my life to take care of children with cancer and their families. It is sometimes devastating, for sure, but it is also tremendously uplifting and fulfilling, and is full of joy and full of hope. One of the amazing things about kids is that, like, kids are full of emotion. They can't contain themselves, right? Like, if you ever watched like a 4-year-old, everything that 4-year-old feels is out there, right?
There's no restraint.
There's no restraint. Even kids who are getting chemotherapy, who are going through radiation, like, they have moments of just pure joy and laughter where they're like running around or, like playing with their new toys or whatever it is. And that resilience or that ebullience is so sustaining. It doesn't in any way detract from how hard it is. Being told that your child has cancer is hands down the most difficult thing that any family ever has to go through. I don't think there's any question of that. And it is that counterpoint, that tension between, the absolute worst thing that's ever happened to your family and these moments of pure unrestrained joy, hope, and happiness that make this such a tremendous honor to be a part of, just to walk with these families along this journey that is so intense, so deeply personal, and so sacred. That's ultimately why I do what I do, and I think that those of us who are called to do this, who are chosen to do this career, that we all feel that.
And what's your outlet? I mean, for me, being here for 20 years now, I've had patients that I've become incredibly close to who, pass away.
Yeah.
I think the outlet for me is going into the garage and maybe banging on the keys or playing music with my kids, as I think you're well aware of. I also do find the research to be sort of a nice outlet.
Mm-hmm.
What are your outlets for all the, angst that you must have over, some of these really, horrific tragedies that we see in the clinic occasionally?
Yeah, that's a phenomenal question. I don't have a good answer for you, to be perfectly honest. I mean, therapy is a really important part of dealing with the psychological, dealing with the grief. I think there's a lot of grief and a lot of sadness. I think we all have our own ways of dealing with that, but it is, as you know, very difficult to figure out how to carry the stories of our patients with us, to respect and honor that without letting it pull us down. And so one of the things that I do is, each patient I've taken care of, many of them give me mementos, pictures, cards and stuff like that. I have those all in my office, and they sit right by my computer monitor so I can see the patients and remember their stories and think about how to do things better in the future. And so I feel in some ways their presence and their strength and their courage and the hope that they put in our team here at City of Hope and in me, in my team, every day when I go in to see the next patient, and that's sustaining.
I love that. And, I love the fact that you brought up therapy. I think it's something that's actually very underutilized in our profession and in many professions in medicine. It takes a lot of courage to bring it up, frankly. But I
think everybody should be in therapy. I mean, I think this is like a baseline thing.
Yeah. Indeed. And just, having that person to talk to, that, sort of ombudsman or, independent party to really just, sort of pour your heart and soul out to. It's so key, and something we don't often, take advantage of in our day to day.
Yeah, no, I completely agree. And then the other thing that you said is the research. I mean, I think that one of the advantages of being a clinician scientist, a physician scientist, is that those things are synergistic, right? The things that you see in the clinic help you figure out how to ask the right questions in the lab, and then vice versa. The things that you learn in the lab you can take back to your patients, right? That's how we got to this clinical trial, which I think has changed so many lives. But that helps a lot for me, right? The pain, the sadness, the frustration that we couldn't do more for each individual child, spurs me to go back in the lab and motivate my people, my scientists to do better, to ask, smarter questions, to get to answers faster so that we can, in 3 years get to a better therapy.
I love this. Pediatric oncology is one of these spaces where, there's just been amazing discoveries early on, I would say near the inception of oncology with multi-drug chemotherapy and so forth. Really transformative outcomes initially with that. There's still certainly a lot to be done. And I will say, I kind of feel like you're at the forefront of it. You, as I mentioned at the outset, run our gene therapy program here at City of Hope. You also run our Alpha Clinic. Alpha Clinic, I think, is a term that's used in a different way at several institutions. Tell us about the City of Hope Alpha Clinic and what that involves.
The fundamental idea, as many of your listeners will know about, is that chimeric antigen receptor T cells, or CAR T cells, are fundamentally this way of taking immune cells, T cells in this case, which are the cells that normally kill viruses, that are virus-infected cells, and reprogramming them to recognize and kill cancer cells. And CAR T-cell technology really revolutionized how we think about and treat hematological malignancies, right? And so this relatively simple idea that your immune system's really good at killing things. Can we repurpose the immune system and retarget it towards cancer cells so that we can really spark an anti-tumor immune response? Has launched this entire field. Now, this is not a new idea. So in many ways, people who do develop clinical cancer, their immune system was unable to stop that. That we call that immune surveillance. And so there was a failure of immune surveillance. So what people who developed chimeric antigen receptor T cells did was to take originally patients' T cells and then reprogram them to recognize specifically things on the cancer cells and then put them back into the body, and then they just go in and search and destroy cancer. As you know, as a solid tumor person yourself, solid tumors are different from leukemia and lymphoma, right? They hide in a specific area of the body. They can weave these sort of cocoons around them. They have their own vascular supply, their own blood supply, and then they can develop these immunosuppressive elements within them that kind of put the immune system to sleep. And so a lot of the work that we're doing now is focused on addressing those components. So we need to get the CAR T cells to the tumor, right? And you can do that a number of different ways. You can inject it directly into the tumor. You can inject it into the blood supply that feeds the tumor. For brain tumors, we inject the CAR T cells into the cerebrospinal fluid, the fluid that bathes the brain and spinal cord. And that's based on work that came out of Christine Brown's lab and Crystal Makel's lab at Stanford, showing that in preclinical models, if you deliver CAR T cells into the CSF, you need about a tenth of the dose that you would need if you were delivering the same CAR T cells intravenously.
And nuanced question here, but do you still need all the same conditioning, chemotherapy, all the pre-treatments ahead of time?
Oh, I love this question.
Yeah.
So do we have like an hour? So this is a very active, ongoing debate. Okay. So we know that for CAR T cells that target leukemia, lymphoma, you absolutely need what's called lymphodepletion, which is chemotherapeutic preconditioning that sort of resets the immune system, clears out some space, allows the CAR T cells to divide, to grow, to proliferate, and not to
gives it some room. Yeah.
Gives it some room, exactly. I don't want to say it's a burning question for me and for the people who do what I do. Like, this is the kind of thing that keeps us up at night.
Yeah.
Is do we need lymphodepletion? Do we need preconditioning for solid tumor targeted CAR T cells? One. And two, does the route of delivery matter? So do we need lymphodepletion for solid tumor targeted CAR T cells if we're delivering them intravenously versus local regionally? Okay, so those are two very open questions. And there is very good preclinical data from Christine Brown's lab, from Saul Priceman's lab, and from other labs that are very convincing, that say that if you provide lymphodepletion for solid tumor targeted CAR T cells, it helps.
Okay.
But A, we don't know if that's true in humans, and B, a lot of those studies were done with systemic delivery, so intravenous delivery of CAR T cells. So lymphodepletion acts to, like you said, clear out, mixed space, but that's mostly in your blood system, so your blood and your bone marrow. So if I'm injecting CAR T cells directly into the tumor itself or into, the CSF, does that really make that much of a difference? One of the amazing things about our ability to do these clinical trials, and also to collaborate with others who are doing similar clinical trials. And here I have to say, pediatric oncology, we talked about this a little bit. It is a bit of a sacred space, in that fortunately not that many kids get cancer. But because of that, we recognize that in order to learn as much as we can, we have to work together, right? So, I have colleagues on speed dial in Seattle, at Stanford, at Texas Children's, at St. Jude. We're talking all the time because we all have sort of parallel trials. And not only we're sending patients back and forth, like if our trial's full, I'll send in Seattle's full, they'll send me, that kind of thing. But also it's kind of like, oh, we saw this thing on our trial. Have you seen anything like that? Mm-hmm. Right. And I got a text the other day that was like, hey, I've been going through our data and I think I see this thing. And I was like, wow, that's super interesting, because I think we might be seeing something similar. And now we're talking about co-publishing, those data so that everybody can learn from it. To answer your question about whether lymphodepletion is helpful and or required for solid tumor targeted CAR T cells delivered either systemically or local regionally, we're all talking all the time to try to answer that question. So like Stanford has a trial where they're treating kids with diffuse intrinsic pontine glioma and diffuse midline glioma. So the DIPG, DMG.
The same two diagnoses you referred to.
Exactly. They had one arm of the trial that included lymphodepletion and systemic delivery. And then another arm of their trial that was intraventricular, so CSF delivery, and initially without lymphodepletion, and then one with lymphodepletion. Seattle has always done intraventricular delivery, so CSF delivery without lymphodepletion. And we've always done intraventricular delivery, so CSF delivery with systemic lymphodepletion, right? So between the three of us, we actually have sort of all of the bases covered.
Right.
And so we've been able to talk to each other and figure out some, I think, really not just scientifically interesting, but clinically important things, right? So what I'll share, and this may be where we're getting out, ahead of over our skis a little bit because it's not published data, but what we're seeing is that lymphodepletion does a couple of really important things, even if you're delivering the CAR T cells directly into the cerebrospinal fluid. So we know that the brain tumor microenvironment, like I said before, really immunosuppressive. And there's a thought that lymphodepletion would reprogram some of that immunosuppressive microenvironment. It's really hard to get serial brain biopsies from patients. And so we can't really go back to the tumor very effectively and say, okay, we gave lymphodepletion, what happened to the tumor? Is it still immunosuppressive? I think the field is moving in that direction, but it's, as you might imagine, it's a bit of a big ask.
It's tricky, isn't it? Yeah. And please, you finish your thought first, and I'll jump in.
We're hopeful that new technologies, like things like, liquid biopsy, will help to answer those questions. But putting that aside for now, none of the trials that currently exist mandate repeated biopsies. So we have proxy measures. We can look at the CSF, we can look at the peripheral blood. Obviously, we see the clinical progression, and we see the MRI imaging to see how these kids are doing. But what we have seen is that in at least three trials, patients who do not receive lymphodepletion are developing anti-CAR immune responses. So we're putting these things, right? So we take the T cells out of the patient, we reprogram them to recognize and kill cancers. We do that by putting a chimeric antigen receptor on the cell surface of the T cells. So that's a receptor that's synthetic, it's engineered. And it's made up of some components that exist in biology, but other things that we invented. And so those things are foreign. And so it turns out that if you don't give lymphodepletion, patients they're recognizing those parts of the construct as foreign, and they're raising the immune response against that. And we think that probably has real consequences. So you're infusing these CAR T cells into patients, but their immune systems are fighting that infusion rather than fighting the cancer. And so that attenuates the efficacy of these CAR T cells, probably. We don't know that for sure yet.
But I think this equates to a pretty major, finding, I would say. It's something that, I personally have struggled with in the renal cell niche, right, in my CAR T trials that I've run, and I would love to say to patients, you don't need to get that chemotherapy, because I do think it really changes the therapeutic index of these compounds entirely.
So are you giving lymphodepletion?
For the most part, yes. For the most part, yes. But we have toyed around with some cohorts without, and I think the dynamics change a little bit.
Yeah, so there's a flip side too, which is that one of the things that we've seen in our trial is that in patients who are not receiving lymphodepletion, when you infuse CAR T cells, you actually entrain endogenous T cells into the CSF. So normally, as you know, you shouldn't have T cells in your CSF, right? If you have T cells in your CSF, you have meningitis, that's a bad thing. They're fighting some sort of infection.
Sure.
But what we're seeing is that over time, as we infuse CAR T cells into the CSF of our patients, they're pulling along normal T cells. Right? And so those things go away if you lymphodeplete. So an open question is what those T cells do. And we have seen that some of those T cells are reactive against the CAR, so that's bad. But we've seen that others of those T cells are not. They're activated, endogenous, killer T cells that are not CAR T cells. They're getting pulled into the mix by the CAR T infusion. And if we could repurpose those cells to kill cancer cells, then we'd be doing ourselves a favor.
That's really interesting. That's right. So, I have to say that there are few labs perhaps as well set up as yours to study these types of phenomena. My understanding through, reviewing your work over the years is that, you really have a beautiful sort of translational setup here where, you're running these trials, you've got the, sort of dynamic biomarkers in patients, and you're able to kind of take those directly to the laboratory and, through both in vitro and vivo modeling, really sort of dissect some of these phenomena. Is that fair?
Yeah, I think that is in my opinion, that's the perfect use case for a physician scientist, right? You're seeing the patients, you're putting them on trial. They are generously sharing all of their data with you that you get to take back to your lab and study to figure out what's going on so that you can figure out how to make better therapies for the next generation of patients, right? And that's super complicated. And it takes a lot of goodwill and a lot of very dedicated, very passionate people across that whole thing. But yeah, I think there are not that many places that are uniquely positioned to do that. City of Hope is one of them. And I feel just incredibly fortunate to be a part of that. I mean, this is something that predated me, will post-date me. This is something that City of Hope has made its name doing for forever. Right? Patient-centered care and research.
It's definitely a neat model. I wonder if you could share with the audience maybe, one of the key findings from your lab within the past 2-3 years or so that really, draws from these clinical experiences that we've had with patients.
Yeah, I think there are a number of things that I think that I'm really proud of my lab for doing. Before I get there, I think there are a number of things that I'm also really proud of City of Hope for enabling our work to do, and here, City of Hope writ large, but also Christine Brown, Steve Foreman, the T-cell folks.
Yeah.
I mentioned that our trial is open at University of Michigan at CHLA, and it's really the only trial of its kind that has that reach, right? So there was another pediatric brain tumor CAR T trial that was open at a number of different centers. That was funded through the Pediatric Brain Tumor Consortium, the PBTC, which was supported by the NIH. Its funding was cut earlier this year, and so those trials all stopped.
Okay.
And so now our trial is the only multi-site CAR T trial for children with brain tumors.
Oh gosh, wow.
Which is a huge problem, because we all recognize that we need to be able to do these things at multiple different places so that we can learn. But even before that, that trial, which was run by Shobha Navai at Texas Children's, that trial was an intravenous trial. So you gave one dose of CAR T cells through the vein. And it's, for technical reasons, much easier logistically to do that kind of a trial, to ship those cells. Our trial is, as we've talked about, delivering the CAR T cells directly into the CSF, and that's a much more challenging logistical trial. So it was a huge and very heavy lift to make that happen for Michigan and CHLA, but we were able to do it. And we did that largely by leaning on the expertise of Seattle Children's, where colleagues had piloted or pioneered some of the technologies that would allow us to do that. Now, they didn't open it, at multiple different sites, but they were able to go to the FDA with some process improvements that we then borrowed in our process, and that allowed us to take the next step and now deliver these cells at a variety of different sites, across the country. So that is something that actually I'm incredibly proud of, because it was a lot of work. It was very difficult to do. And honestly, it's not something that, that doesn't lead to a publication or a grant or a news story, but it is critically important in making these therapies more accessible. And nobody in the Midwest would have been able to get these therapies until, University of Michigan came online, but also really important for how we bring these therapies to the next level. And so our team at City of Hope did it because it was the right thing to do, and you won't see that in, whatever, the New York Times or the Wall Street Journal or the lay press. You won't really even see that in New England Journal or Science or whatever. It's not going to get a podium presentation at ASCO, but super important. I'm really proud of that. That's largely the work of people like Jamie Wagner, who's the regulatory person within what we call the T-cell therapeutics research laboratory. So that's, Steve and Christine's infrastructure. But she worked tirelessly. She and her team continue to work tirelessly behind the scenes to get the kind of stuff done. and you may never hear about it, which is why I wanted to bring it up.
That's awesome. And I assume on top of this like major Herculean, task of delivering cells, on the other side of it, right? By these collaborations, you probably have an opportunity to pull a lot of translational specimens back into your laboratory. Is that right?
Absolutely. Yeah. So one of the great things about our collaboration with the University of Michigan is that has actually spurred a couple of really interesting scientific projects. So the group we're working with in Michigan is one of the investigators, Carl Koshman, who's a really well-known DIPG-DMG sort of biology researcher.
Okay.
Someone in his lab recognized that. So these tumors are driven by a mutation called H3K27M. What that does is it epigenetically deregulates these tumors, and so the chromatin structure changes, which normally, chromatin is open or closed. And if it's open, then it's accessible and you can read it and you can transcribe it and make mRNA, which then turns into protein. But if it's closed, you can't.
Sure.
And what the H3K27M mutation does is it messes with all of that. And so things that are supposed to be open are closed and things that are supposed to be closed are open. And so you end up with this dysregulated transcriptional program that then in part leads to cancer. But one of the things that Carl's group recognized was that this mutation, this H3K27M mutation, it actually upregulated the expression of IL-13 receptor alpha 2, which is the target of our CAR.
Oh, no kidding. Okay.
And we wouldn't have known that had we not started working with them, because they have all this data that they've been working on for a really long time. So they just took a look and they're like, yeah, this is really interesting. And so now we have this tie-in where our labs jointly are working on, what is the linkage between this mutation and upregulation of the target? And can we leverage that in, because Carl's developing therapies like small molecule drugs that target this mutation and the downstream transcriptional consequences of that mutation. And are there ways that we can use that knowledge to increase the expression of our CAR target so that we can be more successful at killing these tumors?
Brilliant. I love that. I mean, it's target validation, right? I mean, you've demonstrated that this HK, presumably histone-related mutation, right, has, kind of driven this abnormality in IL-13 signaling, and boom, you've got a CAR T targeting it, right?
Yeah, so that's really exciting.
I love it. Oh, that's great. See, we probably could do a whole podcast on CAR T. We could do a whole podcast on the Wang family, right? I think we decided we're going to do that at some point. We could probably do another full podcast on gene therapy too, right? So you lead our efforts at City of Hope related to gene therapy. This is something that if you follow biotech, probably one of the most exciting new frontiers, right, in the field of medicine, writ large. Tell us about what City of Hope is doing in the gene therapy space that you're particularly excited about. Just, a couple of highlights.
Yeah. So, I wouldn't say that so I'm the clinical director of our director of our clinical gene therapy program right now. I wouldn't say that I lead gene therapy at City of Hope. I mean, that title really belongs to John Zaya, who for a long time was a mentor and colleague in the Department of Pediatrics, and John, is just a titan in the field of gene therapy. He was one of the first people to work on gene editing for things like HIV and for other infectious diseases like CMV. Collaborated with Steve Foreman for a long time on developing cell therapies to target infectious diseases. John unfortunately passed away not too long ago. By the way, was the director of the Alpha Clinic before me. And so that, he really is, I think, the father of gene therapy at City of Hope. And I am just trying to make him proud, I think. There is a lot. You're right. I mean, gene therapy has been around for a long time, around 50 years clinically. And it's had fits and starts. And, when I was a resident in Philadelphia in pediatrics, I worked with a woman named Kathy High, who was pioneering gene therapy for hemophilia, using adeno-associated viral vectors, so AAVs. And And then when I was a fellow in Boston, I worked with Don Cohn, who's at UCLA, and David Williams, who's at Boston Children's, on a lentiviral-based gene therapy for a disease called X-linked chronic granulomatous disease, XCGD, which is a immunodeficiency in boys. And so it's been, it's been around for a really long time. But you're right that in the past few years, there's really been this explosion of interest in clinically applied translational gene therapy. And that is, I think, due to a number of confluences, right? There's, on the one hand, people like David Liu at the Broad at Harvard have developed these phenomenal new technologies, and Alex Marson at UCSF, of really going in and with unbelievable precision, altering just even a single base, right? You can just edit one thing, which we never used to be able to do before. And so a lot of that's built on CRISPR-Cas9, that came out obviously out of Jen Doudna and Emmanuel Charpentier's work, and Feng Zheng at MIT. But that has become sort of these precise surgical genome editing tools that now you're seeing people really excited about applying in patients. The clearest indication of that, or the clearest use case of that, is in patients with single gene defects, single gene disorders like sickle cell disease, where there's just one base that's wrong, and that causes this terrible, devastating disease called sickle cell disease. And there are lots of different ways to fix that. And fortunately now we have 2 FDA approved gene therapies for sickle cell disease. They actually take very different strategies in how they fix the disorder. So the technology that came out of a company that used to be known as Bluebird Bio and now is known as Genetics is a lentiviral-based technology where they're introducing a full copy of an anti-sickling beta globin into hematopoietic stem and progenitor cells. So those cells, once they turn into red blood cells, they're protected against sickling. But they've introduced a whole new protein, and they use a lentivirus. And that's FDA approved, and it's very, very effective. The other main technology, which is backed by a company called Vertex, and before that, CRISPR-TX, is a CRISPR-based technology with a genome edit. And so they take the hematopoietic stem regenerative cells, the blood-forming stem cells, and they edit very precisely a region of the genome that actually regulates what's called hemoglobin F. And if you do that, you can cause red blood cells in grown-ups to create a lot of this fetal hemoglobin. So normally fetal hemoglobin is produced by babies, infants, and it goes away when you're born. But that hemoglobin F is protective against sickle cell disease. And so if you have high hemoglobin F, generally, even if you have the sickle cell genotype, you don't have clinical signs of disease. There's a whole population of people called HPFH, hereditary persistence of fetal hemoglobin. Those patients generally don't have sickle disease, even if they have the sickle genotype. And so what Vertex figured out was how to edit a particular enhancer region of a gene called BCL11A. And in those patients now, they have much higher levels of hemoglobin F, and so they are also cured of the sickle cell disease. So this is just 2 very, very different ways of getting at the same problem and curing the same disease. And they're both now FDA approved. And I'm very proud to say that our team at City of Hope was able to bring both of those therapies to patients here at City of Hope. We were the first center west of Texas to offer that therapy once it was FDA approved. And we now have a very strong, I would say, explosive program where we're able to cure these patients. And that's led by Michelle Wang in Pediatrics and Pam Becker on the adult side.
I see. I have to tell you, this is something I'm frankly incredibly proud of too. It's interesting. You mentioned CRISPR Therapeutics, working ultimately, the Vertex to develop this technology. I was actually working at CRISPR on their CAR T trials in renal cells. So I saw a lot of these developments happening in parallel. And I did think, gosh, when it comes to implementation, how are we going to make this happen? And then when I heard you share the news internally that we were getting this technology moved along here at City of Hope, it just made me really, really proud to be part of a place that, has early access to these types of technologies.
And I have to tell you, Monty, this again was an example of us, City of Hope, deciding to do the right thing. The sad reality is that these therapies are super expensive. They're very
millions, right?
Yeah,
millions.
It's like 3.1 million for the Bluebird product or the genetics product, and 2.8, I think, for the Vertex product. And it was going to be really difficult to provide those therapies because we have to buy the product. So, that's writing a check for millions of dollars, and then we don't get reimbursed for that, sometimes for months. But, here I have to give credit to Susan Brown when she was here, Kelly Tomlinson, Vince Jensen, Steve Peterson, and Saro Armenian for really just making the case that this was a moral imperative for our patients. And despite the fact that we didn't have a solid business case that was going to make our accountants super happy, they're like, we think that this is a core part of our mission. This is what we need to do. And so they gave us permission, they gave us the green light, and we did it. That's been one of the most gratifying things for me in my career at City of Hope about being here is that, we always put patients first, and we make decisions that allow us, I think, to really push the envelope and do things that are innovative and bold, and right, for patients.
Indeed. I've held you hostage here for a long time. We've covered so many topics. I mean, the gene transfer therapies. We talked about CAR T. Again, I'll lean on the Wang family, who I'm still very intrigued by.
We haven't talked about the Paul family. I mean, I want to hear more about your garage band, literally garage band, right?
Right. Yeah. We're gonna trade places for this on a subsequent podcast and do that. But we always wrap up this podcast by asking our guests what I think is actually a tough question. The title of this podcast is On the Edge of Breakthrough, Voices of Cancer Research. And that term, on the edge of breakthrough, I think it came with a lot of thought. What does that mean to you, being on the edge of breakthrough?
I think there are very few true discontinuities in nature, right? Most of the time, something that you think of as a sharp edge, when you get close enough to it, it's actually not. It's actually a continuous surface, right? And so, you can always take a derivative. It's not a cusp that you can't take a derivative of because there's no slope. It actually is a smooth surface, a continuous surface. And I think that we've talked a lot about things that seem like breakthroughs, seem like cusps. But if you actually are in it, you realize it's been going on for a long time. Gene therapy has been here, been in and out for 50 years. Cell therapy has been going on for decades. So we stand in many ways on the precipice of amazing new innovations, inventions, cures, therapies. But when you actually get into the weeds, you realize the groundwork for these things has been being slowly built over generations, scientific generations, right? Like Steve Foreman, John Zaya, the people who came before them, Jim Allison, they've been doing the work slowly, continuously over decades. And you and I now get to stand on their shoulders and proclaim from the rooftops that we've cured sickle cell disease, right? Which is a breakthrough. It's a revolution.
Yeah.
Right. But that's not an edge. This is something that people have been slowly and laboriously working on it. And Stuart in here would be very upset if he knew that I didn't call him out here. People have laboriously been working on this for entire careers, right? I feel immensely privileged to be able to stand on the edge of what looks like from the outside, what looks like a precipice, right? Looks like we're about to leap into the great beyond, the void of infinite possibility. And also to recognize that that's not what it is at all. This is generations of painstaking work by people who are far smarter, far more accomplished than I am, that I get just to carry across the finish line. This is what we did with the brain tumor CAR T trial, right? I didn't build that technology. Christine and Steve and Mike Jensen, they worked on that for decades before I got here.
Sure.
I just was the person who had the privilege of bringing it into the clinic and the privilege of talking to patients about this revolution, right? Similarly with the sickle cell gene therapy, I didn't build that, but I was able to bring it to patients. I mean, the Alpha Clinic's the same way, we have all these great therapies, and it seems like all of these new things, whether it's in vivo gene therapy or, AI or personal avatars like twinning, all these things seem like cusps, like revolutions, and they are truly revolutions. But it's incredibly humbling for me and grounding to recognize that edge is actually a continuous surface. Does that make sense?
Beautiful. I love that. I'm not supposed to do this, Leo, but if I were to rank all the responses to that question I've gotten on this podcast, that might be the best for so many reasons.
Did you hear that, Dr. Armenian?
You brought in calculus. You're talking about, derivatives, and I was waiting for integrals and law of Laplace and Fourier transforms, but maybe that's for a subsequent episode. Leo,
this is Ross Rockney episode.
Yeah, there you go. We'll bring Russ, our mathematical oncologist, on here. Leo, honestly, tremendous conversation today. Thank you so much for being here, and I hope you'll come back again soon.
It's been an absolute pleasure. But can I flip the script just for a second before we end? I want to. You ask that question all the time of all of your guests.
Yeah,
I've never heard you answer it.
Oh gosh, on the edge of this is tough. You're not supposed to do this, Leo. But what I will say is that, I think that on the edge of breakthrough to me, it just means walking through this campus. Truthfully, as I look to every single laboratory that I pass by, as I look to every single clinic, I'm seeing things that are being done that are going to represent the standard of care tomorrow. Whether it's the IL-13 directed CAR T that you're working on, whether or not it's the stuff that, your wife Stacy's doing in the area of precision oncology to really sort of bring this to every patient, whether it's the stuff that I'm seeing in my own clinics with, standardized targeted therapies for renal cell. I think it's just happening all over the place here. So on the edge of breakthrough to me means, being on the City of Hope campus.
Love it. It's now number 2.
There you go.
Thanks so much.
Thanks for tuning in to On the Edge of Breakthrough. See you next time for more insights from the front lines of cancer research and care.